So I'm here at Benton Elementary, which is a place you're all very familiar with, and a place you might not be familiar with is a science lab on the MU campus. So that's just a few blocks from here, so I'm going to show you where it is and take you there. So if you'll get in my car with me. So here we go. So here we are at the Christopher S. Bond Life Sciences Center. It's a building full of research labs, and my research lab is right up there. The second window, or the fourth window from the left. Now, let's go inside. Welcome to my lab. In here we do a lot of research to study plants and insects. In particular, we're trying to study how one plant called Arabidopsis responds to a little green caterpillar called Pieris rape. And when plants get eaten by caterpillars, they don't just sit there and get eaten. They have a way of responding and driving the caterpillar away. So we try to measure how the plant responds and what the caterpillar does to try to get around that response. But to do that, the first thing we need to be able to do is we need to be able to grow plants, and we need to be able to grow caterpillars so that we always have them available for our experiments. So I'm going to take you downstairs all the way into the basement of this building to show you how we do that. First, we're going to go to the plant room. Hi class, so we're down here in the basement where we grow our plants. Now, the basement, you might not think it's a good place to grow plants because there's no sunlight. But we don't grow our plants in a greenhouse or outside. So to do the kind of experiments we need to do, we grow them in growth chambers, which are special boxes that control the light, temperature, and humidity so the plants grow really evenly the same way every time. So let me take you inside. Oh, and before I do, it's kind of loud in there, so it might be hard to hear me. So these are our growth chambers, and we have several of them. They're all the same, and they allow us to keep the environment the plants grow in really nice for the plants to grow, and it's always the same. So it doesn't get hot during the day or cold at night. It's just always the same temperature, and the lights come on and go off at certain times we specify. This makes all the plants grow really healthy and always the same way. So that we do different experiments, we always have plants that are as similar as we can get them. That way we can compare it to experiments we've done before. So I'll show you the inside of one of these chambers. Up here is the control panel, and this allows us to set the temperature and humidity and have the lights come on however we want. And down here are the different plants we have. So these plants here, they were planted 2 days ago, but they haven't sprouted yet. The idea is to get one plant in each of the pots. These were planted a week ago, and you can see we have some plants sprouting, and the goal is that we'll remove all of these until we just have one plant per pot. These plants have been growing for about a month, and they're just about ready for us to do experiments on them. You can see that they're about as wide as, I don't know, your fingers like this, and they have these older leaves and younger leaves on them, and that'll be important for something I'm going to talk about later. These plants here are over a month old, and they're quite a bit bigger, you can see the leaves get quite a bit larger, but you still have these older leaves coming out, younger leaves coming out on top, and they get older until these oldest leaves are these largest ones here. Now, when the plants grow up and they get fully grown and they want to make seeds, they form a bolt. So this stalk will sprout up out of the plant. The stalk is called a bolt. It'll have some leaves on the side, and on the tip here, you probably can't see it, but I'll show you a picture, are buds where these tiny white flowers will come out. And those will also form into seed pods. And here's a picture of that.
So you weren't able to see in the growth chamber video the flowers of the Arabidopsis very well. That's because the camera couldn't focus on them. But when the plant is ready to make seeds, and there are a lot of different cues which can tell it when it wants to make seeds, it will grow from the base of the plant this bolt, which is a stem that will grow up from the bottom to the top. And you can see that the bolts can branch. So here's one bolt coming up and then it branches off. And you can see there are multiple bolts that can come from the bottom. Down here you see 1, 2, 3 bolts coming off. And at the tips you see these white things, and these are the flowers. They're very small. Here's a close-up view of the flower right here. You can see there are 1, 2, 3, 4 petals. And here in the middle you might be able to see some pollen. After the flower is pollinated, you'll start to see something like this. So this flower has been pollinated and there's this thing growing out of it. This is what has the seeds in it. And it's called a silique, S-I-L-I-Q-U-E. So it's basically a seed pod, just like a bean pod might be full of beans if you've ever seen one of those. And what happens is as the silique grows, eventually these petals will fall off and the silique has all the seeds in it. Here's another picture where you can see several flowers, and some of them have siliques coming out and some don't. So here's a silique, there's a silique, here's one, and there's one. And here are several flowers that maybe haven't been pollinated yet. Eventually the petals will fall off of the flower and all that will be left is the seed pod, the silique. And it will look something like this. Here at the top you can see some flowers which are just starting to grow their siliques, and here you see several large fully grown siliques. Right here you might be able to see one petal that's still stuck onto this one. And as these siliques grow, they will dry out and they will break open really easily to show something like this. So here's a silique that's dried out and is broken open, and what you can see are these little yellow or little orange brown things. What are those? Those are the seeds. So I can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 seeds. And there could be a lot more than that in silique, and there can be a lot of siliques in the plant. One plant could have thousands of seeds. The seeds are very tiny. These guys are about the size of the head of a pin or smaller, like a grain of sand, and that can make them tricky to work with, but after you've been in the lab for a while, you learn some tricks. So I just wanted to show you some close-up pictures of what the flower parts of the Arabidopsis look like.
So you may be wondering how we water these plants. There's no rain down here, and there's no system that makes the water fall down into the growth chamber. So usually when people water, they use a watering can or a sprinkler. We don't use that, one, because we want to make sure all the pots get just the same amount of water, and we also want to make sure that we don't disturb the plants. So if you had like a powerful spray, it could actually tear and injure some of the plants and the things. So we want to figure out a way to water them really gently. So what we do is, we're going to water from the bottom. So all these pots have holes in the bottom of them that water can get through, and we're going to put water in the bottom of this tray and then let it soak up into the soil. To do that, we have a hose. It's just a regular gardening hose, and we have it hooked up to a water supply, and we bring it over here to this empty hole that's in all the trays just for the watering, and we turn it on. You can see there's water coming out, and we're going to let it fill up this tray until the water comes in contact with all the plants. You can see in this empty spot here, there's already water forming. And under this plant, you can see there's water there. So the water will be in contact with those holes, And then the soil will soak it up just like a sponge. You can see here that there's water in the bottom of the tray. So we'll let that sit for about 10 minutes, and then we will take these things off and we'll dump the water out and then put them right back in the growth chamber. But doing it this way, we don't damage any of the plants, and they all get just the same amount of water, just as much as their soil can soak up. So that's how we grow plants down here in the basement away from the sun, and we can grow them all year long. So we'll go back up to the lab and I'll take some of your questions.
So I can take a few questions.
All right, guys, I have this. You're going to pass it around. Okay, but like I said, don't yell into the speaker. Just speak it. Can you hear me right now, Clayton? Mr. C?
Yes, I can.
Okay, so I'm not yelling in the speaker. Is it too loud?
No, it's just perfect.
Okay, so this is how you all should be speaking into the microphone, okay?
Who has any questions? Say, tell Mr. C who you are.
I'm Alice, and how long does it take until about all the pots are filled up with water?
You mean the soil cups?
Yes.
It takes about 10 minutes.
It's Carrington, and what made you want to study plants?
Well, when I first started in college, I wasn't really interested in plants particularly, but I got a job in a research lab, and they studied plants. And so I started working in there for about 3 years, and I got really interested in plants. And then when I finished, and I was going to go to grad school to get my PhD, I found there was a plant lab, and I felt like I already knew a lot about plants. So I just started getting really interested in plants because that's what I had been trained on.
Chris, how do you know that the plants are getting the same amount of water?
Well, we don't know that they're getting exactly the same amount, but we just want to make sure they have more than enough. So we just put the water in the bottom, and then when the soil is fully saturated and it's holding as much water as it can, then we assume the plants have as much as they need. And then we make sure to keep them watered so that they don't ever dry out. So as long as the plants don't run out of water, they should be okay.
This is Diana. How do you how is it like? Oh no, I can't. Is it like the same amount as heat as the room temperature?
The temperature of the growth chambers is set to about what you would think of as room temperature. It's 22 degrees Celsius, which is about 75 degrees Fahrenheit. So it's about the temperature of a comfortable room. But the growth chamber keeps that temperature constant all the time.
This is Aponte. How do you know you have the right analysis?
I'm not sure what you mean. Can you say that again?
How the plants grow? How do you know that they're all growing the same way?
Well, we can look at them and see that all the plants in those trays are about the same size. So that, we can use that to know they're growing about the same way. But if you want to think about the parts of the plant we can't really see, like the chemistry of the plant, or if it has enough of traits that maybe just aren't visible, we can't really know that very well. So what we try to do is minimize that as much as possible by giving them the same temperature, the same amount of light every day, they all stay well watered, they all get the same amount of fertilizer, and that's how we minimize the differences between the plants. But it's not a perfect system.
My name is Robert, and can you show us the experiment with the caterpillar and the plant?
I'll have that here in a video next when I'm going to show you the caterpillars. I'll take one more question.
My name is Derek, and how are you supposed to grow a caterpillar?
Well, I'm glad you asked, Eric, and that's what I'm going to show you next. So I'm going to show you how we grow the caterpillars. Okay?
So we're still here in the basement in our growth chamber room, but we also grow our insects down here, just in another room down the hall. Let's go over there. So this is the insectary where we keep our butterflies. It's just behind this door. Let's go inside. So there are several rooms in here. There's rooms where people grow fruit flies that they use in research, little gnats. There's a room where people have frogs they use to research. And there's also people who have rooms where they keep soybeans and look at soybean pests. But this room is ours. So when we're growing caterpillars that we use for our research, the species we grow, Pieris rape, likes to eat those Arabidopsis plants I showed you earlier. And to grow caterpillars, we have to grow all stages of their life cycle. So we've talked about them before. They start with an egg, and then they make a tiny caterpillar, and then the caterpillar gets bigger and bigger, and then it makes a pupa, and then it hatches out as an adult butterfly. So let's start with the adult butterflies. So right here in this cage, we have a lot of adult butterflies. We have males and females. The males and females differ in the spots they have in their wing. And I'll show you a picture of that here.
So the different genders or sexes of the Pieris rape can be seen by the different spot patterns on their wings. The female has 1, 2 spots on her upper wing, and here on the other side, 1 spot, 2 spots. The male only has 1 spot on his upper wing, 1 and 1. The bottom wing is the same on both. Here he has 1 spot, and she's kind of got 1 spot. So they use these spots to identify each other when they're flying around, and so a male can find a female and vice versa. This is how they find the right partner to mate with.
So they fly around in here and are free to mate with whoever they want. They're not flittering about too much right now. We have, in the cage with them, we have this bottle here, which is a nectar solution. It's honey mixed with some vitamins and diluted with water. And so there's the honey solution in here, and then this is a little piece of cotton that wicks up. The honey will soak up through the cotton, and the butterflies can land right here and take a drink right from that top of that cotton that's soaked in their food. Then we have a plant right here, and this is for them to lay their eggs on. The eggs are just tiny little yellow dots. I'm not sure if you'll be able to see them, but the butterflies lay them all over the plant. This plant has probably 1,000 on it. And I'll show you an up-close picture of the plants and their eggs.
So here is a really up-close shot of that plant I just showed you, and you can see these little yellow dots everywhere. Those are the eggs, and the butterflies, the female butterflies, the mothers, lay these eggs all over the plant. Here's a really up-close picture of the egg, and you can see it's kind of looks like a corn cob, like corn on the cob that you eat. It has some really delicate structures to it. It almost looks like a beautiful building, but this is incredibly tiny.
Go back down here. And then here is a dish of pupa that haven't emerged yet, that will become the adults. Here's an up close of some of those pupa in this dish. They're kind of green or yellow, sometimes they're darker, and these are live pupa that are waiting for a butterfly to come out. I'll show you an up close picture of the pupa. And so let's see what happens when we hatch the eggs on the plant. So in this cage here, we take these plants covered in eggs and we put them in here. And you can see that some of the caterpillars have emerged from the eggs. Now they're too small to see on this camera, but you can tell that several of them have started eating these plants. They started eating the leaves. And all this black stuff you might see is the caterpillar's poop. The scientific word for that is frass. And right here I have a plant. You can see a little closer up. There's caterpillars on these stalks and on these. Now they're green and you might not be able to see them. But if we look at this leaf here, you can see that little chunk that's been taken out of it. That's what the caterpillar is eating already.
So this is the pupa of the Pieris caterpillar. You can see it looks quite a bit different than the caterpillar. It's got this kind of domed shape with these ridges pointing out of it, and this ridge here and this pointy part. Right here is what will become the wing of the caterpillar. You might be able to see some veins of the wing right here. There's also, you might not be able to see, a line of silk coming down here that makes the pupa stick to whatever it's going to grow on. And there's a glob of silk right here on its tail that holds it down. So this is what was the tail of the caterpillar, and this was what was its head, and this will become the head and the tail of the butterfly, and the wings are right here. So the pupa will sit for 3 or 4 days, and then it will emerge as a caterpillar. So here's a close-up view of that plant, and there are actually several caterpillars here. You might not be able to see them because they're green and camouflage, but I'll point out a few. Here's one, a baby caterpillar down there. There's one over here. There are a couple on the stalk here, 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 and here. There's a very small one right there. There's a pretty big one right here. There's another big one right here. And all around, you can see these black specks. Here's a bunch of them. Here's some. There's also some here. And these are the frass. That is their poop. Here's another picture that's closer up. You can see a caterpillar here. It's pretty easy to see. His head's right there. Or her head, maybe. And here's a smaller caterpillar. Here's another one that's kind of curled around this leaf. And there's lots and lots of these black specks. They're frass. That's their poop. And sometimes that's the easiest way to find a caterpillar on a plant, is you see the frass. Here's another close-up pic of the caterpillars on the bolt of the plant, that stem that grows up to make seeds. There's one resting right here, and there's another one up here, and he's actually eating one of the flowers of the plant. They like to eat the flowers too, not just the leaves. The flowers have a lot of nutrients in them that normally would go into the seeds, but this caterpillar is just having a nice meal.
So after we get a lot of caterpillars in here, we might put a few thousand eggs in one cage. But we don't have enough plants to feed all those caterpillars. We feed them via Arabidopsis plants that we grow in our grow chamber. So we take about 50 caterpillars out of here once they get a little bigger, and we transfer them to these other cages. Here's a cage here that has some much larger caterpillars in it. You might be able to see. These are some bigger pots that we grow Arabidopsis in for larger caterpillars. Here's one right here. It's munching on those plants. He's curled up after I picked him up. But I'll show you a close-up picture of what these look like in a second. So these caterpillars grow up in these boxes, and we move them as they get bigger and bigger into other boxes. This one here has some of the biggest caterpillars in it. And also what we have here in the cage is a pupa, right up close. And I'll show you an up-close picture of these too. But the caterpillar will form a pupa right there on the screen, and then what we'll do is we'll take a pair of forceps and we'll remove the pupa very carefully without damaging it. And then we'll take it over and put it right here in this dish full of the other pupa. And then put it right back in the butterfly cage. And then we'll have more butterflies later. And the cycle just goes around and around. This species of butterfly is actually really easy to care for. I wanted to show you the different growth stages of the caterpillar. Right here is a very tiny caterpillar that just hatched a few days ago, and it's what we call first instar. Right here on my second finger is a second instar caterpillar that's probably about 4 days old. Here's a third instar caterpillar that's bigger yet, and it's probably about a week old. And then here's a fourth instar caterpillar that's the biggest kind we have, and it'll make a pupa here in just a few days. The whole life cycle takes about 2 weeks, and then that biggest caterpillar will make a pupa that you can see right here.
Now here's a video I really am excited to show you. This is Pieris rape eating an Arabidopsis plant. We used a special microphone that uses a laser that can actually capture the sound of the Pieris as it eats the plant. Listen carefully and see if you can hear it.
So let's go back up to the lab and I'll answer your questions about butterflies.
Okay, so who's got questions about caterpillars?
How you doing? Sit up.
This is Dejan, and how big can a caterpillar get?
When they're fully grown, they'll be about 2 or 3 inches long. They can get bigger or smaller depending on how good their food is. So if we feed them poor quality food, they won't get very big and they'll make a tiny pupa and they'll make a tiny butterfly. But if we give them really good food, they'll grow really large and they'll make a nice big healthy butterfly.
This is Ayane, and that's the only caterpillars you have. Do you have any others?
That's the only caterpillar that we raise in the lab, but we do use another species of caterpillar, but we buy those from a company because they're hard to grow, and they're called the beet armyworm. The scientific name is Spodoptera exigua, and they eat lots of different kinds of plants. The Pieris that I showed you, they only eat Arabidopsis and other cabbage plants like, well, cabbage and turnip and broccoli. Those are all in the same plant family. The Spodoptera will eat plants from a lot of different families like corn, beets, all different kinds of things. So we can study how the plants respond to caterpillars that are specialized to eat them and how they respond to caterpillars who are not specialized to eat just one kind of plant.
My name is Dwayne, and what's the maximum age of a caterpillar?
Well, from egg to pupa, the caterpillar will take about 2 weeks. So the egg will hatch, and then within maybe just over 2 weeks, about 18 days, it'll make a pupa. The pupa will go for about 4 more days, and then the butterfly can live for around a week or 2 weeks, and then it'll die. So the total lifespan is about a month from egg to a dead butterfly.
This is Nisha. Why do they make that sound when they eat their food?
So they're not really making that sound, but that's just the sound of them crunching up the leaf. They have little hard teeth that are really small, and they just pinch off little sections of the leaf. So it kind of sounds like if you were chewing on a carrot or something, that same crunching sound, that's them eating the plant. I'll take one more question before we go on to the next video.
This is Aviana. How long have you been a scientist?
Well, I started working in a lab right after I started college, which was in 2002. So I've been working in labs, one lab or another, for about 10 years. But I've only been in grad school for, I've been in grad school for about 7 years. So I've been a scientist about 10 years. Okay, so we're going to move on to the next video, and then after that we'll have time for more questions you can ask about anything you've seen so far, okay?
It's recording. So here we are back in the lab, and I'm going to show you how we measure the response of the plant to the insect. Now you may think that plants don't really respond to anything, but they do. Plants respond to insects primarily by making chemicals that will drive the caterpillar away. The caterpillar is eating the plant, and then the plant will sense this and start making chemicals which can poison the caterpillar. Then the caterpillar will be eating and he'll say, I don't like this food so much, and he'll go away. And that way the plant kind of protects itself from the caterpillar. So one of the chemicals, or one of the types of chemicals the plants make is called anthocyanin. So here's our green Arabidopsis plant, just like I showed you downstairs. And green is caused by chlorophyll. But there are other chemicals in here that have different colors. It's just that there's so much chlorophyll you can't see them. Another plant you might be familiar with is a rose. And this rose flower is very red. And this red color is anthocyanins. So the rose puts lots of anthocyanins in its petals, and it puts less chlorophyll there, so you can see all the anthocyanins. And I'm going to show you how we can extract red anthocyanins from Arabidopsis, and then I'll show you how we can get them out of the rose too, and show you something really cool about anthocyanins. So let's take our plant samples over to my lab bench, and I'll show you how we extract the chemicals. So we'll start with the Arabidopsis, and the first thing we have to do is we have to cut off some leaves because we're going to try to measure the chemicals that are in these leaves. And I told you they look green, but there are some red anthocyanins in there. So I'm just going to cut off some leaves, and I'm going to put them right here in this ceramic bowl called a mortar. And we need to grind these leaves up. Basically, we need to make Arabidopsis juice. And you don't make apple juice by just putting an apple in water. You have to grind up the apple and then add water to it and then filter out all the apple chunks, and that's how you get the juice out of the apple. The same way we have to grind the Arabidopsis. Now, if we just started mashing them, they would just kind of mash up. It wouldn't grind really well. So a really good way to grind Arabidopsis is to use liquid nitrogen. So there's some liquid nitrogen in here. It's just going to look like a bunch of smoke, but if you've seen liquid nitrogen before, you know that it freezes things, and it'll make the leaves very hard and crispy, and I'll be able to grind them because they're so hard into a really fine powder. So I'll just pour some of this in here. And I'm going to start crunching these leaves up, and you can see this vapor coming out. That is because the nitrogen here is boiling and it's releasing a lot of cold air as it does that, and that coldness makes the air sort of fog up like this. So right now it's a frozen powder. It's very fine, like baby powder, and I'm going to take a sample of it that I'm going to scoop up here, and I'm going to put it into this test tube. And now I'm going to take this over here, and I'm going to add a chemical to it that is alcohol with a little bit of acid. And this is going to help remove the chlorophyll and the anthocyanins from the leaf. So I'm putting a total of 1 milliliter of this solution in here. And then I'm just going to mix it up. And you can see in the bottom here, it's just some green juice. Not very interesting. I don't see anything that looks red in there. But what we have to do now is we have to let this sit overnight in the refrigerator. And that will give it time for all the chemicals to be removed from the leaf. The little bits of leaf that are still inside here. But to save time, I have already done this. So here's a sample that's been left overnight. It looks a lot browner, and I can see that that it's gotten probably more chemicals extracted from the leaf bits. So now we're going to I'm first going to put on some gloves to keep my hands protected from some of the chemicals I'm going to use. gloves not only protect me from chemicals, but they protect my sample from getting contaminated by the dirt and other things on my hands. So I'm going to take our sample over here and okay. So I'm going to take this sample over here and add 2 more chemicals to it. So the first thing I'm going to add is chloroform, which is a very nasty chemical that can make you very sick if you breathe in the fumes. So we use this special hood that always pulls air inwards. So if we have very smelly chemicals in here that are poisonous, it keeps all of that smelly gas pulled away from us and it shoots it out the top of the building. So I'm going to take a little bit of chloroform and I'm going to add it to my tube. Then I'm going to add some water, plain water. Only this water is very pure and has a lot of stuff removed from it. Then I'm going to shake it up. And I am starting to see the chemicals separate. So I hope you can see this. Adding the chloroform in water makes the chlorophyll move into the bottom of this tube and the anthocyanins, which are pink, move into the top of the tube. So that's because the chloroform doesn't mix with the water. And so the chlorophyll moves into the chloroform and the anthocyanins move into the water that's at the top here. Okay. So now that's how we did it. Now I'm going to show you how we can extract a lot of anthocyanins by using these rose petals that have a lot. So to extract the anthocyanins from the rose petals, and there are a lot of anthocyanins in here, we're going to do the same thing. I'm going to put these rose petals in my liquid nitrogen. And I'm going to take the green part off this flower because I don't want to extract any of the chlorophyll, just the red anthocyanins. So I'm going to put them in here, and then I'm going to crunch them up, just like I did the leaves. So I've ground this up, and what we have now is a lot of powdered rose petals. So you can see it's really pink, just like the petals, and that should be caused by anthocyanins. Actually is caused by anthocyanins. So now I'm just going to try to collect these into this tube. You can see that's all the powdered rose petals. And then I'm going to add some of that alcohol with acid. To it to remove the anthocyanins. I'm going to put a cap on here. I'm going to shake it up. Look how red it is. We're removing a lot of those chemicals. So to remove all the like rose chunks, I'm going to put it in the centrifuge, and it will spin this around really fast, and all the chunks of rose will settle into the bottom, and then all we left at the top is a clear liquid.
Okay, it's recording.
So let's go over to the centrifuge. It's right over here on the other side of our lab. And a centrifuge is just like the spin cycle on a washing machine. You know when your washing machine spins and it pushes all of your clothes and stuff up on the side? What that happens is it pulls all the water out of the clothes because it spins and the water flies outward. This thing works the same way. So it's got these little buckets in here, and we'll put our samples in each side. So this side is just water, but it has the same weight as our rose petal sample, and that'll keep it from being unbalanced. Just like if your washing machine is unbalanced, it can jump. And what'll happen is when I turn it on, it will spin these samples around really fast for 5 minutes, and that will make all these chunks of rose petal fall into the bottom, and then we'll just have our red anthocyanins in the top. Okay, so I think our centrifuge is almost done. I can see right here the speed is coming down, and it should beep in a second to let us know it's finished. Or maybe it doesn't beep, but open it up, and let's pull out our rose sample. And if you can see, a lot of the chunks of rose have fallen into the bottom, and the liquid up top is much clearer. So let's go back over to the bench. And I'm going to use this device to pull out this clear liquid and put it into this small beaker. It's just a little pump, and I can pull this stuff up. This red almost looks like Kool-Aid, but it's definitely not Kool-Aid, into this beaker. Now you might notice that I didn't do something when I extracted from the rose petals. I didn't need to use the chloroform and water because there's not a lot of chlorophyll in the rose petals. They're all red. So all I had to do was use the alcohol acid solution. I don't have to remove the chlorophyll to be able to see this. So we have a lot of this stuff, and it's really pretty red. But what's interesting about this? I'll show you something interesting about it. Okay. So here is some of our rose petal extract, just like we had before, and you see it's very red. Now I'm just going to take a little bit out and put it in this other container. And if you were listening very carefully at the beginning, you'll remember that I said I mixed the rose petal powder we made with a solution of alcohol and acid. So acid has a pH that's very low. If you don't know what pH is, you can look it up or ask your teacher. But basically, acids have a low pH, and the things that have a high pH are bases. If I add a base to this solution, what do you think will happen? Well, I don't know, but let's see. So I'm squirting in a common base known as soap. And what do you see happening? Soap is a base, and what is it doing to the anthocyanins? It's turned them a different color. So I'm going to mix it up a little bit. Now you might not be able to tell what color this is, but I will tell you that it has turned them blue. Why has it turned them blue? Well, it's just how the anthocyanins are made. That when they're acidic, they're red, and when they're basic, they're blue. Now, you might ask, can we turn it back to red? Let's see. I'm going to add some hydrochloric acid, which has a low pH, back, and we'll see what happens. And you'll see it has turned back to red. Now, if you think about this, think about all the flowers you're used to seeing. Most of the flowers you see are red, or they're blue, or they're a mixture of red and blue, which is purple. The only thing it takes for a red flower to become blue is if the plant changes the pH of its petals. And the same thing for a blue plant to turn red. Because the chemicals it's making is the same, and those are anthocyanins. So I've shown you a few things we do here in the lab. We raise plants, we raise caterpillars, and we measure plant chemistry in response to caterpillars. I showed you how you can measure how much anthocyanins are in a plant, which are one of the chemicals plants use to deter caterpillars, and those are the red chemicals in plants. But they can also be blue depending on the pH. So whenever you see a red plant or a red fruit or vegetable like a tomato or a beet or red leaves in the fall, you can know that those chemicals are anthocyanins. But since anthocyanins can be blue, it also counts for blue things like blueberries and all other kinds of blue plants. So I will now take your questions.
Question already. Please put your hand down so everybody can have a turn.
This
Guys, we can't see.
this is Cheyenne. What's a pH?
pH. So I didn't want to explain it in the video. It's kind of complicated and it won't make a lot of sense to you for a few years and you've taken more chemistry classes. But a chemical's pH means how many hydrogen ions it donates to a solution. Now that doesn't make a lot of sense, but when we add the acid to the solution, it will release hydrogen ions, and these ions will bind to other chemicals in the solution. In this case, the anthocyanins. When the anthocyanins receive a hydrogen ion, they turn red. When we add the base takes the hydrogen ion away, and it turns blue.
My name is Trayvon. We was talking about the plants. It sounds different when you with the plants and the plants that we have at our house. Does it seem different when you put water in the soil and then it keeps on growing and growing until it gets old? I think.
Well, plants can grow different ways for a lot of reasons. So, say at your house, if you have some plants, they may look different than the plants at someone else's house. And it's not just because of the water, but it's because of the soil. So the soil could have different nutrients. Some soil is better to grow plants than others. And it's also light is important. So usually when people grow flowers or a garden, they try to grow it on the south side of their house because that gets the most sunlight. But some people don't have southern space. They can grow plants, so they grow them on the north side of the house, which doesn't get as much light, and the plants may not grow as well.
My name is Joey. And what's anthocyanin?
So anthocyanin is a chemical that plants make to produce a color or a pigment. So they want to produce colors to attract bees to pollinate their flowers. Or in the case of roses, we've bred roses for a long time to have really beautiful colors. So it's just a chemical that causes the color itself. So the anthocyanin molecules themselves they're actually red or blue colored depending on the pH. But whenever you see a red or blue flower that's caused by the same kinds of anthocyanin chemicals, it's just that the plant will have them in a high pH or low pH environment to cause the different colors. If you see a purple plant, that's red and blue mixed together, and that's caused by the same thing.
Have you ever had trouble with the plant machines? Like, have you ever had trouble with, like, the plants growing?
Oh yes, we certainly have. Those machines are really complicated, and they work kind of like a refrigerator. When it gets too hot in there, they cool, and then when it gets too cold, they heat it up, and they break down fairly often. And we luckily have associated with our lab some great guys who know how to fix these kinds of things. So if you're interested in science, you don't have to be a scientist. All these machines we have people who are really smart who know how to fix them and come and repair them when they break. And they're very expensive, complicated machines, so those people need a lot of training and need to know a lot about science to know how to fix them.
Does it take a lot of chemicals to change the colors of of the colors of the water and stuff?
So it doesn't take a lot to change the color of the anthocyanins. They're pretty sensitive to the pH, but it depends on what concentration you're using. So when I squirted in the soap, that was just regular dish soap we use in the lab, and I didn't need a lot of it to change the pH. But when I added the acid to turn it back red, I had to add even more acid to compensate for the soap. And that acid solution was actually very concentrated. But you can make the acid solution weaker by mixing it with water, and then you would have to add more.
All right, Mr. C, I think we have 3 more questions here.
All right.
My name's Kayla, and on the caterpillar video, why do you only take care of just one kind?
The reason we just grow that one kind of caterpillar is because they're really easy to take care of, and that's the kind we use the most. The other kind we can just order from a company, and for $50 they'll send us 1,000 eggs that we can raise. But that species of caterpillar is kind of hard to care for because you have to sort of trick them into coming out of their pupa by exposing them to cold temperatures so they think it's winter, and you have to feed them different kinds of food that we don't have as available. So they're just more complicated to grow, but we can order them whenever we need them. You can also order the Pieris, but they're so easy to care for that it's cheaper for us just to raise them ourselves. So after the egg hatches, it'll take about 2.5 weeks for it to turn into a pupa. Then the pupa will take about 3 or 4 days to emerge into a butterfly.
Is there any other colors that on the chemicals?
There actually are. So the anthocyanins are responsible for red and blue coloration in most plants, but there are other colors like you see orange carrots and the green, of course, that you see in leaves. So the green is caused by chlorophyll, which is a green color that is what helps the plant absorb light and use that energy to grow. The orange color in carrots is caused by carotenoids, which are just another kind of chemical. And when you see like a bird, like a robin, that has a red breast on it, or a cardinal that's all red, they actually get that red color from the carotenoids from the seeds and plants they eat. So they use the plant's pigment from their diet to make their colors bright. And that's important because if the males are really healthy and have really bright colors, the females will know that they're very good males to have their babies with. If the males are really sick, they won't be able to use those colors as well, and they won't be as bright. So the females will know that they're not as healthy and they won't make as good of children.
All right, I think that's all the questions we have. That's all the questions we have, guys?
Thank you.
The class, they want to say something to you. Guys, the microphone's over here, so you got to say it this way. Thank you, Mr. C. We really appreciate it.
Thank you all so much.